in the dark.
Okay, guys. Sorry for the delay. A few minutes. So I noticed there's less people here than normal, not by much, but I can see the drop off is starting. I mean, maybe they're just late.
It's the snow.
It's the snow. I heard we might have an inch tomorrow.
Really?
And I heard that upstairs. Okay, so we're starting. Ooh, someone just put lotion on.
Is it too strong?
No, it's fine. Smell is such an amazing thing. It takes a while. Only when it's good. Sometimes it's really bad. Anyways, okay, so exams back on Thursday, hopefully. Fingers crossed. I've heard from some people they felt it was really easy. Some people were crying. Someone told me people were crying. Don't let my class bring you to tears. It's not worth it. Remember, you have your health. Right? You have your health. Okay, so we're starting to talk about cell cycle. For those who took 408, a lot of this is repeat. Because what I'm going to do is I'm using cell cycle as the backdrop for starting to talk about DNA replication and mitosis and the mechanisms behind that. And so in order to talk about those things, I need to explain a little bit of cell cycle regulation. So that doesn't mean if you took 408, don't come to class. That means just feel good about the fact that you might know what I'm talking about right away. And then that's the same for those of you who will take 408 with me in the fall. You'll get kind of a little head start on that. Okay. So I just like immunofluorescence, which is why I put in this picture. This is showing the different phases of the cell cycle. You guys all learned that cell cycle of mitosis, and cell cycle in general. So you've got interphase, prophase, metaphase, anaphase, telophase, late telophase, and then cytokinesis will be next. And I just like to show you immunofluorescence just because it's a really cool way to look at things. So in this case, this was from a paper where they were studying the role of SUMO, which stands for small ubiquitin-like modifier. So it's a ubiquitin-like protein that's also post-translationally conjugated to other proteins and affects their function in some way. So you could think when something is simulated, it's some sort of regulation of the function of that protein. You can't see?
No.
What should I move? Should I move that?
It's over.
Should I move myself? So you've got SUMO 1 and then SUMO 2, 3. Don't worry about the detail of what SUMO is doing. This is just kind of a figure that I can use to introduce the topic. So here in interphase, there's a few things you can notice. One is you can notice the staining of SUMO. So everywhere, so green is SUMO, blue is DAPI. Everywhere that it's green is where SUMO, the SUMO that they're telling you about, is. And blue is DAPI, which stains DNA, so that's always the DNA of the cell. And so you can see that SUMO seems to be an interphase for whatever reason, localized. SUMO 1 seems to be localized around what? What do you think that is? Nuclear envelope, something like that. And then things start to reorganize as you enter mitosis, right? And so SUMO 2, 3 also kind of appears to be excluded from the nucleus during interphase, and then is somehow involved with mitosis. You guys all agree with me? Somehow. The DNA, what do you guys notice about the DNA as you go from interphase to mitosis? What is happening? Someone raise your hand. Huh? The DNA is dividing, but what do you think is happening to the chromatin? Yeah.
It looks like it's condensing.
It looks like it's condensing, exactly. So here you've got this kind of like amorphous mass of DNA, and then it starts to get organized here, metaphase, and then you have anaphase. And so, but it's from this kind of large mass to this condensed structure, exactly. And then you can also see that there's staining here going on. There's some sort of interaction between SUMO2-3 and the DNA in some way, chromosomes, right? Because you can see here it seems to be overlapping constantly with the DNA, so it's binding somewhere on the chromosome. It's probably involved in some way with chromosome segregation, regulation. SUMO1 is not, right? It actually seems to be involved with, what do you think? What's in between the DNA there? Well, so that's interesting that you're saying that. So this is the whole DNA of the cell. So you can imagine you would actually have a bunch of chromosomes lined up here. Would you say? I heard someone say something. So probably mitotic spindle here, right? Yeah. So it's probably somehow involved in function of the mitotic spindle. So there's a lot you can learn by looking at these images. But really, I'm just showing it to you because I think it's pretty. And I think it's a good way to introduce a new topic. Anyways, okay, onward. So don't freak out because you're like, what was she talking about? Really, it's just start to get used to looking at these things and thinking about things in a more kind of inquiry-based way. Okay. So what is cell cycle? Basically, its function is just to control copying of the genome and organelles, and then distributing it to the other cells. That's what you're doing over and over and over again. Right? You've got your genome, and then it gets copied during S phase, and then it gets separated into two different cells, and then the cells divide. Right? So S phase and then mitosis. And so obviously this is critical for survival of an organism because this is growth, right? And yeah, it's needed for growth and to replace cells that die every day. So the major elements of cell cycle and what we're using this as kind of a jumping off point for is S phase, which is DNA synthesis, right? DNA replication. And M phase, mitosis. So towards the end of interphase, so there was a few years ago when I taught this, there was a lot of confusion about what interphase was. And I'm just going to take this moment to just clarify. You guys might already know this, but interphase is not a part of mitosis. It's the part of the cell cycle that is not mitosis. Okay, so there's always someone who thinks it's like the first part of mitosis. It's not. There's always someone who thinks that it's a separate part of the cell cycle from S phase. No, actually, S phase happens during interphase. Okay, so just think of the cell cycle as M phase and interphase, and towards the end of interphase, you have S phase, or DNA replication. This might seem obvious. Hopefully it seems obvious to most of you. So DNA is replicated towards the end of interphase during S phase, and if all goes well, right? If the DNA replicates properly, there's no DNA damage that caused replication to stall. Then the cell progresses to M phase, and that's where the duplicated chromosomes are segregated to the two daughter cells, and that's mitosis. So this is the cell cycle figure you see over and over again in every class you take probably, right? It looks like a clock. And so it's showing that there's these gap phases, right? So you've got G1, which is one gap phase. You've got S phase, G2, and M phase. What's the purpose of having these gap phases. Yes?
Your checkpoints, so, help make sure that nothing, goes bad.
Okay, so you use the word checkpoints. So I want to, so that's good because we're going to talk about checkpoints. But I always want to be careful. I feel like I'm constantly telling students in my labs, be very careful about the words they use because sometimes in science they mean something else. So yes, they are a period in time where the cell's kind of making sure everything's right and repairing whatever, right? And sometimes checkpoints can occur during your gap phases, but there are also checkpoints that occur during mitosis. So that's just like a language thing. But yes, it is a period of time during cell cycle where the cell's kind of evaluating whether or not things have occurred properly and whether you can move on to the next phase. So in G1, so did someone else want to add? I think, sorry, you were going to add.
I would say it's like the same thing.
Same thing. So what's happening in G1? We're preparing for S phase, and so we want to kind of make sure, this first of all, what you want to just make sure the environment is appropriate so that you can move on into S phase, right? And then in G2, it's important for the cell to be able to make sure, not like it's like a being that thinks, but to be able to kind of ensure that S phase occurred properly and finished, right? Because you want to go into mitosis if you're still replicating your DNA? No, right? And then also one thing that needs to happen during G2 is the cell needs to grow because remember it's going to split in two. So it kind of needs to double its size so it can split into two. So that needs to happen. And then there's this G0 that people have probably told you about, right? Who heard about some G0 phase? Okay, good. You guys have all been here. Okay. What's G0?
It's like the rest phase, so if it isn't working proper, it'll go to G0, and if it doesn't get fixed, it'll G destroy.
So it's like, yeah, it's like a rest stop on the highway that is the cell cycle. Yeah, so if something's not working properly, it's a time for the cell to repair. Okay. Well, does anyone want to add anything about G0? Yep.
I thought G0 was for some kind of cells like neurons or like muscle cells that don't replicate.
Yeah, so also cells that don't replicate are permanently in G0. Yeah, what else? Any other information? I'm just going to have you guys teach the class. I probably could do that. I could probably put up a topic and we can discuss and somehow I could pull out all the information from the whole group. Should we just convert to that style?
You have to split yourself.
You just want me to, what'd you say I have to share
You have
myself?
to share your salary.
Hi. You'll be so sad when you see how little you'll be like, peanut butter and jelly for everyone. Yeah. Sorry, I was just lamenting for a minute. I had a friend in grad school who went in, she was from Singapore. She's been to Singapore. Okay, good. We've got somebody who's been to Singapore. So she went into research because in Singapore it's a very lucrative career. And so she was in it for the money. That was all she was in it for. And so she came to America and was studying to be a researcher. And I remember one day, she said, I forgot what she said, but my boss said to her, wait a minute, why are you doing research if you want to make money? Like, why are you doing that? And she was like, well, that's not one of the highest paying careers where I come from. And we were all like, what? Like, because no one else goes to research to be rich. You go in because you love asking questions and are not satisfied with whatever's just placed in front of you. But anyways, she went into venture capital. And she's rich now.
She didn't go back.
She did go back.
Yeah.
So she's like doubly rich. Anyways, actually I don't know, I haven't talked to her. She might be completely poor, but I have a feeling. She was very driven, so I have a feeling she's rich. Anyways, she is not in G0. Okay. So cell cycle, let's go back. rein myself in. Cell cycle is tightly regulated, right? So I love to talk about regulation, and with everything in the cell, we always have to question how is it regulated. So it's controlled by a number of proteins that act as biochemical switches that only allow progression to the next phase of the cell cycle if the previous phase was properly completed. And so this brings up checkpoints specifically. So cell cycle progression is regulated at specific checkpoints. At these checkpoints, the cell checks for certain things, and if those things aren't appropriate, then cell cycle arrest happens. So you've got your, in late G1, you've got your start checkpoint, which is just the cell kind of checking is the environment favorable, or are we under conditions of starvation, what's going on here. If all is well, do we have horrible DNA damage? That'll slow things down, right? And if all is well, then you go into S phase. There's another checkpoint at G2M, so that's right before entering M phase. So that's that G2M transition. And there you're asking, is the DNA replicated? And again, is the environment favorable? If the DNA was not replicated, sometimes there could be DNA damage that might cause a stalled replication for example, or sometimes something else could have gone wrong that prevented appropriate DNA replication, and you want to stop the cell cycle from progressing in that situation. And then there's the metaphase to anaphase transition, and that's smack in the middle of M phase. It's not in a gap phase. And so that's why I pointed out they always have to be careful with your wording, because checkpoints very much refer to these 3 checkpoints. And in going from metaphase to anaphase, the real question is are your chromosomes attached to the mitotic spindle? Because should you go from metaphase to anaphase if your chromosomes are not attached to your mitotic spindle? Probably not, because metaphase, remember, is when all your chromosomes are lined up, and anaphase is when they separate. And if you're not attached to mitotic spindle, then the chromosomes will not be distributed equally to daughter cells. Yes?
Is G0 when we're stuck at a checkpoint, or is it some other point in the cycle that we're not seeing?
G0 is actually before you go into S phase specifically. Yeah. So when you're stuck at a checkpoint, that's usually, it could be anywhere. I guess you can say if you're stuck at start, maybe that would be similar to G0. But you can have checkpoints that will halt the cell cycle in the middle of it. And so it's really, the question is, are you going through cell cycle or not? If not, if you're like a neuron or you're not dividing, then you're not even going into S phase, and that would be G0. Okay? Yes? Someone?
So if the cell is not going to replicate, why would it invest the energy of starting the process?
If the cell, okay, so the question was, if the cell is not going to replicate, are you talking about any type of cell, or are you
No, I'm
talking
talking like, let's say for a neuron,
for neurons? Okay. Then why would it invest the energy of,
getting to G0.
okay, remember G0 is not, you're not getting anywhere, you're just there.
But then the cell grew.
Yeah,
inside.
so like, if you think about like your brain, for example. See, I feel like I'm going to go into an area that I'm not an expert in here, but if you think about, for example, your brain, like, so there's cellular differentiation that happens where you go from like this kind of pluripotent cell into differentiated cell, and then you assume your job. And depending on the type of cell you become, you'll follow different programs. And so some cells, like neurons, don't divide. So they're in G0. But they're there because you need them to survive. The organism would not exist without, right? So it kind of, before it became a differentiated neuron, it divided up to a certain point,
That's what I'm saying.
and then it's there. But if you don't have neurons, you have an organ. Like if you have a mammal, for example,
No, I mean, like, when everything is well and done, why do cells keep on, the totipotent cells keep on dividing and stop at G0 because it's gonna be a neuron and it's not gonna go anywhere above that, so why waste the energy? It doesn't seem to me like it's efficient.
Okay, so the question is why waste the energy to become a cell that's never going to divide again? Well, I guess the other side would be what the answer to that question is more wrapped around in why don't neurons divide, right? Or why don't some cells divide? And so if we understand that, then you can think about it. Like, so what's the purpose of each cell type? And why do some divide and some don't? And that, I think, is beyond what I can answer at this exact moment, but it's definitely worth chatting about. I'm not going to make an extra credit because sometimes it's just like a very simple question with a simple answer, but this starts to become almost philosophical. And I don't know that I want like essays dropping in my inbox. But we should definitely talk about this more at my office hours, and everyone is welcome to come and give me their input. Are you satisfied?
Can I ask? Isn't it like G0 for neuron? They are not dividing, but they are doing their job.
Yeah.
That's it. It's not, they are not using any energy or they are not actually going through anything basically.
You're just an interface.
Yeah. So the neuron is there, but for the other cells, when they are dividing and they have some problem, like their DNA is not duplicated correctly, they send G0 and cell repairs it, or it is not able to do it, just goes opposite, goes and dies.
Right. Okay, so did that help you? Yes.
Also, so you can go with like a liver cell that stays in G0 until it's damaged, and then it can repair itself up to what, 3/4? If at least a quarter is removed, it can actually repair itself. So you can actually go back through the cell cycle and replicate it when needed. Otherwise, if it stays in G0, you just
Well, that is the other thing. G0 is not a permanent place for everything. It's all situation dependent. So don't think of G0 as a permanent place.
For nuance, it's permanent.
Yeah.
Huh?
Nuance is permanent after a certain age.
For neurons, well, but neurons can regenerate. So,
isn't it, because she was saying something about why would you waste the energy and all that, and I will ask the other way, why would you waste the energy to divide if you didn't have to produce anything new to substitute? Is it
okay.
something, yeah, like, it's just like a switch, if you don't need it, it's like with your clothes, if you have, your clothes are okay, you don't have to buy a new one.
Okay, guys, let's. So this is actually generating a really interesting conversation. So sometimes with such a large class, what happens is if an interesting conversation starts off, some people aren't engaged by it and start talking. And that makes it very difficult to engage everyone, re-engage everyone. But yeah, so what you just brought up is just like you're thinking of it as why waste the energy to divide to get to a certain point. Well, why waste the energy to divide when you don't need to divide?
Like, divide only when it's damaged and really fix it.
So it depends on the cell type. So, but we should continue talking about this because it's interesting. I think it's interesting for a lot of people. But I'm going to keep moving in the interest of covering material. Okay, so cell cycle is highly regulated, controlled by many different proteins. So why is it so important to regulate the cell cycle? Cancer. Because if you don't regulate the cell cycle and you have unregulated cell growth, you could have cancer. Yep, that's a big one. So then that brings up how do cell biologists study cell cycle and cell cycle regulation. So I'll go through a few different things just to kind of give you guys an idea of the type of methods people use to study. The first thing to understand is that the machinery that's involved in cell cycle regulation is highly conserved throughout eukaryotes. And that's why you have all these model organisms that people use. So a lot of the research that led to the current understanding of cell cycle was done in model systems, mainly yeast, different animal embryos, and culture mammalian cells. There's a ton of stuff that was done in yeast, which is why so many of the proteins are called CDC, because it stands for cell defective cycle or something like that. And so there were all these yeast mutants that were generated and characterized using these kind of complementation assays and conditional mutants, and they were basically just numbered. And so as you'll notice, when we study cell cycle and all the proteins that are involved in regulation, they're always called CDC and then a number, because they were just numbered. Okay, so yeast is a very commonly used model system for cell cycle regulation, and there's two types, fission yeast and budding yeast, and it really has to do with how they divide. And so this brings in the concept of temperature sensitive or conditional mutants. So there is a question on the quiz about that, so hopefully you guys all understand that concept now. But just to review for those who might still be confused about it, these are genetic mutants that are fine at the permissive temperature, but at the restrictive temperature, which is usually when you increase the temperature, then they show their mutant phenotype. So in this example, it's showing cells that at the normal temperature are dividing. So it's showing a population of cells. Remember, if you have a whole population of cells, they're not all at the same phase of the cell cycle unless you synchronize them. One might be at G1, some might be at S phase. They're all at different phases of the cell cycle. So you have them kind of just going through cell cycle. But when you change the temperature to a high temperature, which is known as the restrictive temperature, then the defect will be there, and they'll all kind of keep cycling until they reach their point of defect. And in this case, something having to do probably with starting, the start is probably defective, and you're stuck at between G1 and S. Okay? And so you might say, well, what does temperature have to do with it? Well, you can think of it in the sense of there's probably some mutation, and at a normal temperature, the protein folds just fine. But at a slightly elevated temperature, the protein that has the defect is more likely to be misfolded and not function properly. That's the easiest way to understand it. I think at some point, someone taught me temperature. Oh, I think your umbrella is in my office, by the way. That's nice. Sorry. But wait one second. Brain not working well. So Noah explained to me why the temperature was so important, and I remember just being like, okay, there's this restrictive and permissive, and I don't get it, and why does this work this way? And I think the easiest way to understand it has to do with the stability of a protein that has a minor mutation in it. If it's a very, very horrible mutation, you'll never even get that gene product. But if it's this kind of minor defect that's fine at one temperature, but if you raise it a little bit, it misfolds, and that's a really nice system for studying things. And you might ask me, well, why do we need this? Well, remember, these are cell cycle mutants, and if you have a mutation in a protein involved in the cell cycle, can you grow those cells? No, like, if you didn't have this temperature sensitive thing, and you just had a mutation in some cells that was involved in allowing cell cycle to progress, would you be able to grow those cells? No, because there's some kind of mutation that's preventing cell cycle. So this allows you to propagate cells that have a mutant, yet cells that have a mutation, yet still study that specific gene. Okay? Yes?
So I'm still a little confused. So the permissive temperature, it has the mutant, but it's showing it?
They all have, so the cell always has the mutation in it. The question is whether it's acting wild type or acting mutant. Okay? So at the permissive temperature, it's permissive for growth. That means cell cycle is happening normally, which means that the mutation's not showing. Because if it was showing, it would progress normally. Make sense? Whereas at the restrictive temperature, it's restrictive for cell growth. So that protein is probably misfolded or something, so it's not going to function properly. So cell cycle is going to halt. Does that make sense? Yeah. So actually, people always get confused with permissive and restrictive, and I just want you to think about it like in terms of permissive for cell growth. Is cell growth permitted or is it restricted? Yeah.
Okay, so my question is, was this helpful for studying the cell cycle? Because did they all stop at the same place and then they realized, in terms of the mutants, did they all stop at the same place? And then they realized, okay, this must be a checkpoint, or how is this?
Yeah, so by using this to study cell cycle, it's useful because you can visualize cells and tell which parts of the cell cycle they're in fairly easily, because they're either, they either look interphase, or they might look metaphase, or they might look anaphase, or they might look like cytokinesis has occurred. You know what I'm saying? So there's very easy to visualize, and so there are many different mutants, not just one, right? And they're gonna, depending on where that gene, which gene is mutated, it'll have a different phenotype. And so that's why they were able to use these mutants to study cell cycle, because you say, this, I'm going to give some examples. You'll see that there's some kind of defect in a protein required for cytokinesis, and then, which is what I'm about to show you actually, and then you'll see that the cells at the restricted temperature did not divide. Okay, so I'll show you this right now. Okay, so this is using budding yeast. I heard that all the way up here. Amazing. So this is using budding yeast to study defects. So remember, budding yeast buds. It creates a little bud rather than just using fission. So wild type, so this is where they had a temperature-sensitive mutant of CDC15. Okay? So there's some mutation in the gene for CDC15. At the permissive temperature, it functions just fine. The protein works fine. You can grow the cells. They go through cell cycle normally. At the restrictive temperature, CDC15 misfolds or whatever and doesn't function properly, and you can see the phenotype. So if that CDC15 is no longer functional. Okay, and so you can see with wild type, cells are kind of at different phases of the cell cycle. This one is starting to bud, this one has a little bud, this one has a bigger bud. That's that. If you look at the restrictive temperature, what do you guys see? Okay, yep. So they all seem to be in the same step of budding. Okay, so what did I heard something else down here.
Cytokinesis, so that's part of separating.
Stuck at cytokinesis, right, because they're not completely separating. Right, so if I asked you which point in the cell cycle is affected, what might you say? Yeah, so maybe they're having these cells have a hard time exiting mitosis. Would you agree with me? Do you think S phase is affected? Probably not, because it's gotten this far, right? Right. So that's exactly how these mutants work. You would, so you do further studies on, okay, I think this is involved in exiting mitosis. I think this is involved in cytokinesis. You do a certain number of studies. You publish your paper. You get your PhD, and then you move on with your life. Yes.
Does it affect is it affecting the gene or the protein, like the temper?
So you're wondering what this mutant is about. Okay, so when you talk about a mutant, usually it's affecting the gene, but remember the gene encodes a protein, right? So if there's one nucleotide change for some reason in the gene, then that might result in one amino acid being different in the protein, which might not be enough to really affect the folding normally, but maybe if you increase the heat a little bit by going from 37 degrees to 42 degrees Celsius, then that protein suddenly will misfold.
So like the heat could disrupt the bonds.
It's not, yes, you're asking what the heat does. The heat is really just destabilizing something that's barely stable as it is. So it's not a general misfolding of proteins because, you know, if you heat shock an organism, it is going to destabilize proteins if you go high enough in temperature. In this situation, the restrictive temperature is not so high to denature all the proteins in the cell. It's just denaturing a protein that was barely holding its shape as it is due to its mutation. Okay? Any other questions about this concept? I almost always have some sort of question on an exam about temperature sensitive mutants or something like that. So it's definitely important to make sure you understand this. Yeah.
So wild type can grow like in a certain rate of temperature, while mutant can only grow like either below or above that range.
So don't think of it as wild type and mutant. I made a mistake. I should not have written wild type. I should have written permissive. Okay? Think of it in terms of permissive and restrictive because it's one cell line, one mutant cell line, which grows normally at the permissive temperature and doesn't grow anymore and has its defect at the restrictive temperature. Okay?
But wild type will grow regardless whether it's.
Yeah, wild type always grows because there's no mutation. So this is wrong. When you guys go over your notes, change this right here to permissive. Okay? So that's what happens when I write too fast. It has a wild type phenotype. Yeah.
With chemotherapy drugs and things like that, and what cancer cells in general, is there any research being done where you can somehow bring the cancer cells up to a specific temperature, and then at one point they'll stop working, or is there anything like that, or is there hypothetically could that be done?
Well, so there's a small problem with cancer, which is that it's usually within an organism that you don't want to kill, right? So that's one problem. But so you're asking, is there a way to harness the fact that certain cells could be killed if there's some sort of mutation in them specifically? And that's pretty much not all of, but a lot of newer cancer drugs target specific proteins and specific mutations. And so you could say that that's what people are doing now. They're not using temperature because that's fairly non-specific, but maybe they'll target a gene that's overexpressed, and that overexpression is required for tumor cell growth. And by targeting that gene, they can slow tumor cell growth. Yeah.
For the restrictive cell, how are you supposed to recognize which phase it's in?
For the restrictive cell, how do you recognize what phase it's in? Well, so you would have to be comparing to your permissive, right? And so if you look at your permissive and you know it's budding yeast, right? Which means that, if you go back to this, you know that budding yeast is going to look like this. Normal, starting a bud tip, larger bud, 2 cells, right? You know that that's how it looks. And so if you look here and you look at this, you can say, okay, starting a bud tip, larger bud. You see that there's different phases of cell cycle occurring here, and then you see these all look the same. And by seeing that, so that means that there's a block somewhere. They all progress until they hit the block. So you can imagine like a whole bunch of people running and hitting the wall. You know? Yeah. Does that help? Yeah. And then you have to identify in your head, like, okay, what phase of the cell cycle might that be? And then you kind of go back and think to yourself, okay, so we've got S phase and M phase. Okay. It's not S phase because it looks like it's something having to do mitosis. And then you think of the phases of mitosis and think, okay, well, it looks like having a hard time with that final separation. What's that called? Probably cytokinesis. So I'm asking you to apply everything you know. Yeah. Okay, any other questions? I saw another hand up. No. Good. What we're going to do, yeah?
I was just gonna make sure that we're all on the same page for the parts is what do you think CDC 15 does?
So what do you think CDC 15 does?
I know, is it a cell cycle regulator?
Well, it's clearly a cell cycle regulator. You're absolutely right because cell cycle arrests when it's mutated. But we want more specific than that, right? So what phase of the cell cycle is being affected? Right, so cytokinesis specifically. So you would say a cell cycle regulator involved in accomplishing cytokinesis, or a cell cycle regulator involved in exiting mitosis. Okay? Okay. So that's also another thing that I think can be very hard in this class is trying to figure out exactly how much mechanistic detail I want. I would say the more mechanistic detail, the better, because just saying a cell cycle regulator doesn't tell me that you truly have thought deeply about it, and I know you can. Okay. So here's a practice problem for you guys to work on right now. So here you're using temperature-sensitive mutants to assign gene function again, and I'm going to tell you what's going on here. First of all, so cells were grown at the permissive temperature and then shifted to the restrictive temperature, so you know what that means, and allowed to grow for 2 hours. And then I'm asking here, what is the function of CDC 15? Now, is that the same? Huh, that's so funny. But anyways, don't worry about it, because actually here, all you know is it's involved in cytokinesis, right? Here we're going to go a little deeper. So what we're looking at here is microscopy, where they're staining for different things. So here's the stain, DAPI, all the way down. These are all restrictive temperature you're looking at. You're not looking at any permissive right here, only restrictive. And then here, you're staining for actin. All the way down, okay? And so here's different mutants that they're screening. CDC7, CDC11, CDC14, and CDC15, okay? And so what you want to do is look at the DAPI, which remember stains DNA. Look at where it is in each cell type. And then also look at actin and try to remember what actin does in cell cycle. If you can, it might look like something specific in these images. And you can imagine that since we're asking about CDC15, that's where your defect is, so you want to zero in on this lower square here. And you can kind of imagine that this is more what wild type looks like. CDC7 is more what wild type looks like. So go ahead and talk to your friends, if you can. And I'm going to come visit and clarify any questions. But I really want to know what's the function of CDC15, and it can be as simple as it's a gene regulatory protein or a cell cycle protein involved in blah. Okay?
No, if you talk about actin, that's what gives rise to endoplasm. So, right, because if you can't, right, so you have your centromere, then you have your part of it, you have your microtubules, then they go back and that's what you're doing. It's all about shortening. The DNA is actually fine. Remember that it divides, and it divides, and
That's CDC.
To the left side is one, right? The right one. But both of them are readers. But I think it's single vibration. Because if there was a single vibration, you don't have to the DNA to move to the opposite side. It's just that you can't cut that big cell. I mean, it's still, you have to remember that it's a big cell, and all you try to do is just put it inside. Yeah, I think it's not. It's yeah, I think it's microphone was a good writing. I think that's what.
So guys, I'm gonna give you guys two pieces of information that might help. One is that this is fission yeast, so it should be dividing down the middle. It's not budding yeast. And the other is that actin, why do we care about actin? Actin in cell cycle is involved in formation of the contractile ring, which is involved in completion of cytokinesis. So that also might help.
If it's something that test, you have to say, so many other things.
Microfilament is the whole structure of
Okay, guys. So, based on looking at CDC7, CDC11, what do you think that, what's going on with the actin in these cells that are behaving more normal? They're getting ready to divide because all the actin is building up in the middle, right? And so that's going to form the contractile ring, which is that contracts and causes the pinching off of the two cells from each other in cytokinesis. What do you guys see in CDC15? There's nothing. So CDC15 must have something to do with reorganization of actin at a certain location in the cell that will form the contractile ring, right? And so if you put those two together, here you're already seeing there's some defect in cytokinesis. And then here you're seeing there's some defect with relocalizing actin. Yeah.
How can you tell that it has something to do with the organization of the actin? Like, isn't the actin part highlighted, but there's nothing highlighted on that, so how can you?
Exactly. So sometimes it's the absence of something that tells you something. So normally it should all be in the middle. And so you have at this phase of the cell cycle, you have reorganization of actin to form the contractile ring. And the next step, if you could look later, would be cytokinesis. But here you don't have that. So the mutation results in a lack of reorganization of actin into the contractile ring. A lack of recruitment of actin to the plane where the contractile ring will form. So you have to think backwards in a way, right?
Yes, so then in other words, since the rest of the CDCs have actin and they're showing it, then it's not a mutated function of those ones?
Exactly. The other CDCs are showing that the actin is organizing to form the contractile ring. These are mutants, but they're not mutants in this. Yeah. And so what they were doing here is they were screening a whole bunch of them, and they identified CDC15 as having the defect because it's not happening there, and these are behaving normal, like wild type. Now you might, if you look very closely, you might see that there might be some defects. Where did I say? No, I take that back. No, you can't tell anything about the other ones. Yes?
So in the DAPI state, what are the little arrows in the lower left pointing out?
So I think that this is, I'll have to go back and look at the paper, but I think that this was showing that you're going to notice that your nuclei aren't equally, so with this temperature sensitive mutant, you usually have arrest of cell cycle at a certain point because they kind of all progress until the defect. But here you don't you don't really have that because the nuclei in this cell there's 2 on either side. Over here there's 2 on either side, but then over here, it's like they get pushed apart by, I think, actin also. No, I think it's a tubulin thing. I'll have to go back and remember exactly what pushes them apart in bacteria. I mean in yeast. I'm sorry, I was thinking about bacteria for a minute.
So do you think they
Mitotic spindle. But yeah, so I think they must be adjusting that, but I don't pay attention to it for this purpose. Who is just saying something? I just heard something. Oh, you? Huh?
So are they all on the same stage level, or are they on a different level? They were arrested during a different phase.
They've all arrested in the sense that they couldn't form their contractile ring. So they're arrested prior to cytokinesis.
I was just asking about the left.
But it's the same cell.
Okay.
Yeah, so this is actually, yes, sorry, I did not teach this here. But so usually the way this is done is when they've got cells next to each other like this, these are the same exact pictures stained for different things. So these are just stained for DAPI and these are just stained for actin. Okay, yes?
So the actin is supposed to be in the middle.
Yeah.
So then why in CDC11 is it shown up on the polls?
So, right, so that's assuming that they are all, dividing or all at the same phase of the cell cycle. That's not always the case. So in this case, some are in the middle and some are not. Probably, I'm guessing these cells just weren't at that point yet. Because usually when they're all at the same phase, there's something wrong. Unless it says in the methods that they synchronized them, but these they were not synchronized. Any other questions? You guys are good though. You're picking out details that I'm trying not to get you guys to focus on. That means but that means that you're looking the way you should be looking. Okay, are we ready to move on? Okay, so immortalized cell lines can grow indefinitely, and they're another tool for analyzing cell cycle. Oh my God, we are not moving fast enough today. And let me take that back. Immortalized cell lines are not only used for studying cell cycle, you can use them for everything. So cell culture, tissue culture cells. These cells grow forever, usually because they're transformed in some way. Either they're a cancer cell or they have some kind of viral DNA integrated into them. Monoclonal antibodies. You can use hybridoma to make monoclonal antibodies, but that's a different story. But immortalized cells, so yeah, you can use immortalized cells to make hybridoma, which is when you fuse a cell to a B cell to make it kind of immortal and secreting the antibody of your desire for the rest of the cell's existence. Yes. So some tools for studying cell cycle in cell culture are direct visualization, just looking at the cells, right? So cells in mitosis are really easy to visualize, right? They're either rounded. Cytokinesis is easily visualized, right? That's just looking at the cell undergoing cytokinesis. There's no staining, no nothing. You can just tell by looking at it. Or you can use different DNA stains or antibodies. So over here, you're looking at 1, 2, 3, 4, 5, 6, 7 cells that are all sitting together in cell culture. And there are 2 antibodies being used here. One is a green one that's against tubulin to stain the microtubules. And then the other is the DNA stain, DAPI, to look at the DNA. And so what can we say about this picture in terms of cell cycle? Does anyone have any observations? Yes?
It looks like the cell in the center right is going through mitosis in the metaphase.
Center right, this cell is going through mitosis metaphase. Do people agree? Why do you say that?
All the DNA is all compacted in a line. The tubules are trying to help separate the sister chromosomes.
Right, so microtubules are set up into mitotic spindle. The chromosomes are condensed and lined up on the very stereotypical metaphase plate. What else did people say? Yes?
There's some cells that are in interphase as well.
There are cells that are in interphase. Which ones? What do their nuclei look like?
Well, it's not condensed, so all the cells are kind of bluish purplish.
Okay, so the DNA is not condensed. The cells look the nuclei look kind of bluish purplish. What else? What can you say about the microtubules?
Oh, I don't see any.
Okay, so all of this green is all microtubules. And so what you learn in cell biology, but I'm not teaching you now, is that microtubules are organized very differently in interphase versus mitosis. In interphase, they're organized at the, where? Centrosome. At the centrosome. And that centrosome is usually located near the nucleus, and the microtubules kind of just radiate out in every direction from there, and they're used as these tracks to move things around in the cell. But during mitosis, the microtubules reorganize because the centrosome duplicates and becomes each pole of the mitotic spindle. And so you can see that those microtubules and these microtubules look very different. What do you think is going on over here? What phase of cell cycle is this? Huh? Prophase. Right, protophase is before you have duplication of your mitotic spindle, or you're just starting to, but they're not actually separated yet. But you do have condensed chromosomes.
But there's still some attached.
So you can learn a lot from just looking at pictures. I meant that to sound more educated. You can learn a lot from looking at pictures. Okay. Oh, do you want to go back?
But you're talking about, like, it doesn't look like prophase like pharma. It's just like trying to explode. It's not like.
Well, so remember, those are chromosomes, right? And there are a lot of chromosomes, and those are the chromosomes themselves are condensing, but before they condense, after they condense, then they line up. And that would be metaphase. Okay. So you can just look at yourself, right? Also, there are ways to tell whether cells are in S phase. And you can do that by using a base analog. Do you guys know what a base analog is? Do you know what an analog is? And we're not talking about like electronics. Yes.
Just a compliment.
A complement. Not a complement, but similar. Yeah.
It's structurally very similar, except for maybe one difference, and then they function the same. So the analog is the
Right. So if you compare an analog to the original compound, they're structurally similar. Maybe one small change in the structure, but the function is the same. Exactly. A lot of the times when you make drugs, if you're a chemist and you're synthesizing drugs, you'll synthesize many analogs, and you're looking for a drug that's going to have the same function, but little adding a side chain here and there might increase its effectiveness in some way or its whatever. So this analog is a base analog, so it's an analog of a base. It's called BRDU, and it's a thymidine analog. And so it would be used instead of NETs when doing DNA replication. So usually what you do is so this is just kind of showing sequence, and what would happen is your BRDU would be incorporated anywhere a T should be incorporated. And then you can use an antibody that will recognize BRDU and be fluorescent, so you can stain it. And you can stain the cells that have incorporated your base analog. Why do you care about this? Well, what happens during DNA replication? What happens in terms of bases that are present in the cell? Where do they go? If you just have random nucleotides sitting in your cell, do they stay just sitting there, or do they get incorporated into something during S phase? Right, so you're making more DNA, so those bases are being incorporated into the new DNA. Right? Remember, DNA replication, you got to get your bases from somewhere. I know it's crazy. So during S phase, you're going to incorporate all this BRDU into the cell, so all the newly made DNA is going to have BRDU. And then it will be detected by an antibody against BRDU. And so this figure here, which is right out of the textbook, so if you don't get it, feel free to go back and look at the figure, read the figure legend, read what's around it, and see if you can understand it more deeply. But these are epithelial cells in a zebrafish gut, and all the green cells are labeled with anti-BRDU. And then they did a counter staining for all the cells, probably actin or something. And those are all red. And so all those yellow are BRDU positive cells. So what can you say? If a cell is BRDU positive, what phase of the cell cycle has it recently gone through? S phase. So it's important to understand this because I might at some point give you an exam question that says blah, 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 drug-treated cells, blah, blah, blah. These cells are BRDU positive. And so if you can understand BRDU, base analog incorporated into newly synthesized DNA, that can be detected. If you have BRDU incorporated into DNA, it must be newly made, right? Because how's it going to get incorporated into DNA that's already existing? It won't. Any questions about this concept? Yes.
So just increase the concentration of BRDU base compared to like regular thymine.
Well, there won't be any thymine put in, and there'll be BRDU put in. So what you do, so good. I'm going to tell you the protocol for how you do this in the lab in hopes that it will make more sense. So I just got a question about that made me think that explaining the protocol might simplify things. So forget about zebrafish, just think cell culture, okay? And you're studying yourself. What you're going to do is you've got your cells growing on a tissue culture plate with media. You're going to add your BRDU to your media. So it's present, it's available as cells go through cell cycle. And then you can treat your cells however you want, and then you're looking to see whether or not cells have gone through cell cycle. So you'll have cells that are treated with some drug or not. And then you'll harvest them, or you won't harvest them, you'll stain them for anti-BRDU. And you can then look to see how your drug affected cell cycle because cells that are very high, very BRD positive, will have just gone through cell cycle or S phase, whereas cells with no BRDU positive cells, something probably affected going through S phase because they weren't able to replicate their DNA. Okay? Yep.
So would it depend on the type of drug that you're using and what you want to do to those?
Yeah, so yeah, this is exactly. So this is only a method that can be used to answer any question where your question is, does this affect S phase? So it could be simple as screening drugs, or it could be looking at effects of some protein on something, or it could be looking at infection with a virus or bacteria or whatever. But as long as your question is S phase affected, then you can use this assay. And so that's why I'm introducing it, because it's an assay that researchers use to study cell cycle. It's important to be able to have assays for different parts of cell cycle so you can start to think about which phase is affected. You know what I should do is I should put together a really nice practice problem for Thursday where you can assess different phases of the cell cycle using different methods. Because we already spoke a little bit about conditional mutants, we spoke a little bit about using antibodies, about visualizing cells. You can start to see where they are in mitosis. You've got this to see whether they've gone through S phase. It's a lot of tools in your toolbox. Okay, so let's see. So I'm going to introduce flow cytometry, and then we will probably have to go home. So who knows what flow cytometry is? Yes. Who took Michelle Snyder? Okay, so then you guys know. She goes over that a little? Okay. That was about 4 of you. So, and then those of you who had me may have seen it, but a lot of you guys don't know. So basically, it's just a way for counting cells based on whatever property. So this is just showing each of these little dots, you can imagine, is a cell. And so you pour your cells into this machine. You don't really pour them in, they get sucked up, but that's a different story. Pour them into this machine, and they kind of drop through one by one in front of a laser. And based on what you tell that laser to measure, it will count them and measure them based on whatever. So in this case, if you just want to count cells, you could use flow cytometry to count your cells. It's going to make this single stream of cells, and every time a cell passes in front of a laser, it's going to alter what the laser does. And so then it's going to be detected, and the computer's going to go 1, 2, 3, and so on. And so you'll count however many cells, and then you can do other things too, like measure how the light scatters from the laser, right? So if you have me, and then you bring in my daughter, who's about this tall, and you shine a big light at us, will we have the same shadow? No. And so it kind of works using the same concept that if you shine a light at a big cell, and you shine a light at a little cell, it's going to scatter the light differently. And then you can start to group cells into different populations. Now you might be saying to me, what? Now this requires you to think for a minute about the fact that different cells have different shapes and sizes, right? So for example, if you take all the cells out of your blood, well, minus the red blood cells, so all your white blood cells, and you run them through a flow cytometer, and you look at the side scatter and forward scatter, and that's just 2 measures of how these lasers bounce off the cells, they'll cluster. And so all the cells that are this shape will cluster in this. And so each little dot represents where one cell falls in the plot of this arbitrary measuring. So this is side scatter by forward scatter. So all of these cells here are one type of cells. All these cells here are another type of cells. All these cells here are another type of cells. And they're all going to just scatter be plotted based on their size. So that's one thing you can count them. You can separate them based on their shape and size. You can also use antibodies and analyze them that way. So if you use an antibody against CD4, which is a self-surface protein, then you can say, pick up all the cell, computer, detect all cells that are positive for CD4 or CD8 or CD3. So in this case, they're looking at, first they found all their lymphocytes because they're CD45 positive. So they used an antibody in their cell with their cells to stain all the CD45 positive cells, and then looked at their side scatter. And so they got these two populations. They've got lymphocytes, which is T and B cells, and then they start to look even more specifically, right? So then they've got your CD3 positive cells, which is again another marker where they're able to look at CD3 or CD4. Basically, flow cytometry is a way to count cells based on whatever you choose. We're running out of time. If you have to run, go. If you have questions, feel free to come up. I just realized we're over time. We have so much fun in this one. And I'll start with this next time.